And what we've been talking about is the main job that these organs are doing, like the testis is carrying out spermatogenesis. And the ovary is helping to carry out oogenesis. But there's other things that these two organs do that we haven't really covered. I'm going to talk a little about it today. The testis is also in charge of secreting a really strong steroid hormone called testosterone. That has an effect on embryogenesis. And the ovary is in charge of secreting another steroid hormone called estrogen. And these hormones affect the morphological formation of the embryo to give us either a female phenotype, male phenotype. Now, most people would say, well, what gives you the female phenotype? You know, it starts out at the level of the chromosome, XX chromosome. You have two X chromosomes that usually leads to the female phenotype. You have an X and a Y that usually leads to the male phenotype, the male morphological anatomy. But just having those chromosomes doesn't guarantee that. There's a process, and the process is driven by endocrine hormones working on morphology. All right, so we see this in many different animals across the vertebrate range, across invertebrates as well. So the end product of this is something that we call sexual dimorphism. That is, not only is there a difference between organisms, but I know this you're gonna feel like duh, but I mean there's a difference between the genders in one organism. That seems kind of obvious to us, but it's also kind of weird to have one organism with two different architectures, right? One organism with two different architectures. How do we get those two different shapes? It's this process of sexual dimorphism. In some animals, it's not so obvious. Some animals, you have to open them up to tell the difference. Like, we were talking about a redfish today. You have to sort of be real familiar with the redfish to know the difference between the boy and the girl. When you open up a redfish and you clean the redfish and you open up, you can tell the difference between whether they have an ovary or a testis. That's real obvious. If you look on the outside, sometimes the boy might be a little more red. The girl might be a little bigger because she's holding eggs. But sometimes it's pretty hard to tell the difference between the boys and girls. Okay? So sometimes a year sexual dimorphism is strong. The external difference. Sometimes a year it's not so strong. And there's other animals where it's pretty obvious, like the greenhead mallard, right? Greenhead, the mallard duck, greenhead mallard drake has this very green head that's almost luminescent. It shines. It's pretty easy to tell the difference between that and the hen. He's made to almost blend into grass, like almost to not be noticed. It's pretty weird. In the same organism, one sex wants to be noticed, the other sex doesn't want to be noticed. It's not necessarily because she doesn't want to be noticed by him, but she's trying to hide from predators, right? It makes sense to you probably. The mother is often trying to hide. Why do the males want to be seen? So in most vertebrates, the males are more decorative. And it's because they vie for attention from the female. The female chooses. Okay. But what are the males doing in this situation? Those male greenheads, they're doing two things. One is they're competing with other males, trying to say, I got this part of the bank. They're also trying to attract a female. So this sexual dimorphism leads to another type of selection that we haven't talked about yet, right? We talked about natural selection. Natural selection is the most obvious kind of evolutionary mechanism. Natural selection was the thing that was proposed by Darwin and his younger colleague, Alfred Russel Wallace. Together they communicated this together, even though Darwin was writing about it in his own notes a little earlier. And their mechanism, natural selection, is what we accept now as, they got it pretty right, the mechanism of how evolution works. But there's also other mechanisms that can happen. Sexual dimorphism, there you can recognize that another pressure is sexual selection. Even inside an organism, there's selection going on. And this leads to some weird things. Like we grew up thinking about an elk or a deer or a buck having big horns. Well, that's normal. But if you think about it from an evolutionary point of view, it's kind of weird for an elk to have such a huge rack. It's so big, it's hard to hold up. It's easy to get caught in the woods and in briars and stuff. It's almost a liability. Why would an animal have this huge big thing on its head? It almost doesn't make sense, but it's the power of sexual selection. For some reason, elks with bigger racks reproduced more. So the tendency to have a bigger antler rack was selected for. So they get bigger and bigger. Right? Sometimes it's just subtle, sometimes it's more obvious. Like with blue jays. Pretty hard to tell the difference between a boy and a girl blue jay. They look the same. But like with a cardinal. The cardinal, the boy is very red, and the girl, you can tell the difference. Her color is less red. Some birds, you can hardly tell that they're in the same organism because their colors are so different. See that with fish too. You have fish in the hallway right now. That's Spongulus grandis, the cockahoe minnow, where the boy has silver specks, like polka dots. And the girl is the exact same shape, but she's just slate gray. Right. You see this in vertebrates. You also see it in invertebrates. Sexual selection. So what that means is it's been around a long time. You see it in vertebrates and invertebrates. It means it's a pretty strong, kind of like it's a process that's been around in biology for a long time. But what I'd like for us to do today is I'd like for us to look at two different possibilities of sexual selection. I'd like for us to look at whether there's a sexual dimorphism in a physiological characteristic, and a sexual dimorphism and an anatomical characteristic. So to look at an anatomical version of sexual selection, we're going to use an invertebrate organism. That means what we're doing today could be called comparative physiology, or even part of this is invertebrate physiology. So This is an animal that occurs on the coast of Louisiana. You can buy them, or if you have an instructor who likes to go to the marshes of Cocodrie and collect his own animals for free, then you can get this special species because it's hard to buy this exact species. So other scientists call this the fiddler crab. Fiddler crabs occur all over the world. They are known for making burrows, and they're known for this specific extreme type of sexual dimorphism. That is, the boys have this very large cheliped or claw. They're called fiddler crabs because it's almost like he's carrying a fiddle. Right? That's the boy. On the other hand, the girl has two regular sized claws. That gives you an example of that. If the girl can get along and do fine and eat with two claws that are the same size, regular size, what is so important about the boy to make him grow that huge big claw? And what's so important about it is that the females pick males that have larger claws. In the same way that I was talking about a while ago, what are the males doing? The males with that large claw, they are signaling to other males. There's a territorial signal. If you watch the marsh for a little while, not without making a little noise, a lot of times these will come out of the grass. And the boys spend like all day lifting up their arm like this. And they're telling the other boy to stay a certain distance away. If you watch long enough, you'll see that fiddler crabs tend to stay about this far away from each other in a marsh. Not only are they separating themselves from the other males, they're also trying to attract a female to come down into their burrow. And over millions of years, you know, you could kind of do it with a slightly bigger claw. Over millions of years with fiddler crab, that claw has just gotten bigger and bigger and bigger because it's selected for bigger and bigger and leads to more reproduction. So that sexual selection is a pretty strong thing. But what I would like for us to do is to compare an anatomical sexual dimorphism in crabs with the physiological sexual dimorphism in humans. That means we're going to use live animals today. But before we use live animals, I'd like for us to all be on the same page about the 3 R's of animal usage. I do not really think about the 3 R's every day. But some of this gives you a tool. If you have to describe to somebody in your circle of friends might say, well, why don't you use live animals? You don't need to use live animals anymore since we have computers. Well, it might be true. You can get a lot done with computers. There's a group of us at Nichols that feel like the only way to understand living organisms is to work with living organisms. Right? So I've preserved this lab with the experience of us touching some living organisms to make sure that you've had some experience with living organisms. On the other hand, the 3 R's give us at least something to consider whenever we use living organisms. This is the 3 R's of animal use in biology. So before we use animals, we always consider a way that we can reduce the number that we're using. If possible. If If we were doing an experiment today that I wanted to publish, we would use 100 fiddler crabs. We're not trying to publish this. I'm just trying to illustrate something to you. So we're going to use about 20 today. But whenever possible, you reduce the number of animal written. Whenever possible, refine the technique to avoid change. That means you keep the animals as comfortable as possible. So these animals were collected this weekend. They are being kept at 5 parts per thousand cylinders, which is kind of similar to what they were sitting at in the marsh. Right? They could tolerate something saltier, like 30 parts per thousand. That's old seawater. But 5 parts per thousand is really where they're found in the marsh. They're found on the fresher side of an estuarine marsh. We're feeding them fish flake, keeping them hydrated. Sometimes with animals, if you're gonna do something to them that might be stressful with it, something like a fiddler crab, you can put them in the refrigerator to cool them down so they feel less pain. Or with a fish, we could sedate them with MS-222, a sedative. We're not going to use that today, but we always think about it. Final thing we think about is, well, maybe we can replace the organism with something else. Something that's sentient. It's like feeling or thinking. Instead of using humans for this part of the experiment, we're using fiddler crabs. Instead of using fiddler crabs, we could use somehow, we could maybe use something like a fiddler crab model. Instead of using a fiddler crab model, we could use something like a computer simulation. Instead of using a computer simulation, we could use something like pictures. To get data like we did today in the past, I've seen physiology students where to measure the size of stuff, and measure something as boring as bolts and screws. Play around with it. Yeah. Measure the mass of a bolt. Okay, that gives you a number. It's so crazy boring, there's no way I want to have a physiology student look at bolts and screws. Right? So we consider if we can reduce the number, yes. Refine our technique to avoid playing as much as possible. We're going to be careful. Are we going to replace? No, we're not going to do this one. Sometimes we will, but today we're not. We're using fiddler crabs. Right? These fiddler crabs that we're using today allow you to say that you've worked with an invertebrate species. So that's phylum Arthropoda. Who's in the arthropods? That's the insects, crustaceans, spiders, right? This is a crustacean that we're working with. Crabs and crawfish and shrimp, they're all called decapod crustaceans. Decapod crustaceans. Gabby, you know what it means to be a decapod? Kind of in the word.
Wait, can you repeat it?
You know what it means to be a decapod? Like us, we're bipedal. Bipedal. Decapod. 10 legs. Crawfish have 10 legs. Lobsters have 10 legs. Crabs have 10 legs. Shrimp have 10 legs. Fiddler crabs have 10 legs. You'll notice that they got 4 walking legs on each side. And then they got 2 cotyledons, which is like the grabbing legs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's their 10 legs. The animal that we're using today is named Eucaminax. red fiddler. It was collected over in Louisiana. I had to go down this haunted road. I see some Operousas. You still see, you know where I was? Where? What do we call that road? Yeah, I guess I'll call it that. The Mighty Sally Road. Mighty Sally Road. Sounds like a girl's name is Sally. But that's Sally, which means like salty body. Mighty Sally Road. 7. The road that goes from Copa Rica to that. Okay. So to do this experiment today, we're going to get some data. The data is going to be part of our first lab report. It means we're going to analyze the data. Before you do an experiment, and in your lab report, when you do something like this, you need to be able to state our objective. I'd like for you to think about our fiddler crab's objective. The objective of our experiment. This is not the same term as the object of the game. It's not the object, it's the objective. So write that whole word out in your notes. What's the objective of our study? What the objective of our study is not to prove anything. We're not trying to prove something. As scientists in biology, we gather evidence and make interpretations. In other disciplines, you can prove it. We already use the word prove in biology. We're not trying to show something. If your objective is to show a change, then that represents some bias. What I would like for you to say, your objective. My objective was to test whether, then we say the thing we're going to measure. In this case, it's mass. To test whether the mass of female fiddler crabs is different than the mass of male fiddler crabs. To be an objective that gets full credit, don't say prove, don't say show it. You don't say it's your objective. And my objective was to show that female fiddler crabs are smaller than males. Okay? That shows some bias as well. You don't want to say which one you're even guessing. You see here how vague this is. All it says is I'm going to test whether there's a difference. That means even if there is a difference or there's not a difference, I can still fulfill my objective. Get it done. I said here it was to show that female crabs are smaller than males. If I didn't get that, then I would say, well, I failed my objective. So you don't want to set yourself up for that. Once you have your objective, you can kind of modify these terms into a null hypothesis. Now, the null hypothesis is different from your hypothesis. Hypothesis is sort of like what you guess. We use the term hypothesis pretty often. My theory, my hypothesis, get a third COVID vaccination before the end of the year. Something like that. Okay, that's our hypothesis. The null hypothesis is something different. It's almost like a mathematical statement. Because in the null hypothesis, you always use the terms not different. No matter what you expect, when you state your null hypothesis, you always say something is not different from something. Like this. This is my objective. My null hypothesis is, my null hypothesis is that kilogram not different. Once you state your null hypothesis, that is kind of required for you to use statistics. Then you can do statistical analysis. And after the statistical analysis is looked at, you can make an interpretation. And what is not the interpretation is that it's wrong or right. Right. But this is right. If there's no hypothesis. Okay. We don't say our null hypothesis was wrong or right. And it don't even sound right to me for us to say the null hypothesis is true or false. Don't say that either. After statistical analysis, we say whether we accept that null hypothesis or we reject it. Good null hypothesis the way it stands. Then we make an interpretation and we accept or reject. I'd like you to be thinking about that today. This is an anatomical objective. I would like for us to do a physiological version of this. The sexual dimorphism goes into physiology as well. Like, where should we see sexual dimorphism in physiology? Like, if we could take blood and measure estrogen and testosterone, we would expect to find testosterone in males, estrogen in females. That would be a physiological sexual dimorphic character. But does it mean everything is different? The physiological parameter that I would like for us to test today in humans is heart rate. Is heart rate different between males and females? Heart rate. It's a physiological characteristic. Renee, what do you think? Can you create an objective that kind of matches this? Kind of like my crab objective?
I would say,
Look at the words from that crab one and just fill in the blanks.
I would say my objective was to test whether the heart rate was different between them.
I agree. There's something about the term between that it gets me all mixed up. So it's kind of boring, but the way that I would say it compared to what you said is to test whether heart rate in males is different from heart rate in females. Ms. Rachel, can you turn what Renee said into the null hypothesis? Give it to us.
Yes, the mass of, I was reading the thing. The heart rate of males is not different than the heart rate in females.
I agree. That's a good null hypothesis. We have an objective and a null hypothesis in two different systems. One's an anatomical, one is physiological. One is crustacean, one is a chordate. That's our phylum. Homo sapiens. Sometimes people forget the name of our species. We are home station. People think, we're just talking about singular, and we leave off the S, but no, even me, one person, I am home safety. That's gonna be our other animal that we're gonna do an experiment on today. So in order to do the first portion of the fiddler crab, we need somebody to be able to do the measurement of mass using a balance. We need somebody to take the data down on a notebook, not on notebook data recorder. We need somebody to take images on their cell phone of the action. For that, somebody on the computer up here to type in the data. We need somebody to be the crab handler. Who wants to not touch crabs and be a data person? Okay, you got it. Remind me that. That's Miss Danielle. Okay, you got a notebook, Andy? Okay, who wants to use their cell phone and take images of the people doing the work? Okay, who wants to work up here on the computer and type in the numbers? Okay, who wants to touch the crabs? Okay, what's your name again? Mr. Robert? You gotta have a helper. Yeah. Okay? Yeah. Need somebody to work on the balance. How many names? Men's and a young man?
Yes, sir.
Okay. Need somebody to use their cell phone and take images. Ms. Courtney? Yeah, let's do this out the top. Need somebody to work up here. Is that you, Mr. Correa? You gonna work on the computer up here? Mr. Allen? Yes. Who else? All right. Who else? We're going to talk about in the meantime, I'd like for everybody to turn the page on their notebook and make an image of our animal model here. Okay, the reason that you're drawing our crab is that it'd be easy for your lab report for you to go to Google and Google up the map and get images of the crab. But I don't want to get Google images of the crab. I want these to be images of the crab that you drew, like Aristotle did. Some of what we do in biology has been done for thousands of years, and you can do it that way. Right? So even though a fiddler crab is small like this, I don't want you to draw it small like this on your paper. Like Mason, how big do you think it should be on your notebook page? Yeah, it should kind of fill up the page. Like you were doing a portrait of a fiddler crab. Then you can take a shot of that portrait and then you can put that in your lab report whenever you describe the methods. Yes, ma'am. Yeah. That is called the telopid. You call it a claw? That's how you spell telopid right there. Okay, just like the blue crab also has a difference in an abdomen. Right? The underneath side. Now see the male abdomen right here. The abdomen of the male is kind of skinny, kind of like the Washington Monument. Whereas the female's abdomen, like the blue crab, is kind of big like this. We tend to call it an apron. Try to get the nail in position first. All right. All right. You can do it, its eyes and its eye stalks. You can kind of get its legs. You can get its claw. You can sort of see the teeth on its claw. Got some little holes down here. You can kind of get the shape of this big claw, this big teeth there. Now, as it moves around, it's just like too bad. Just like if we were out in the marsh, you just have to go with it. Animals don't always just sit down for us to draw them. You gotta try to follow them if you can. Okay, well, I'm ready for the folks that are helping do some of the work of the data. You want me into that? I need crab handlers. And data takers and the people with cell phones. Let me get a little holder for the crab. I'm not ready for the first crab yet. Balance is made so that everyone cares. Numbers. There's going to be a little bit of wet. Before you take the next piece. We want photos of crabs, we want photos of people too. So Mr. Allen, I'm going to need you in about 5 minutes after we get the data from there. Okay, I'll try to get the face here for you. That's a pretty good face right there. Okay, so you had it on one page. I'm going to make you turn the page and now do it again. The way that you study an animal is you don't just do one image. Take it once and then with a slightly different direction, slightly different pose, you do it again. And then you do it again and you do it again. After 3 of them, you start to get some of the elements of it, like the hairs on the legs, and exactly what his mouthpieces look like. This guy's name for it is Red Hinge Fiddler. You can see he's got some little red marks right here at the hinges of his heel pads. You can see the little tubercles, which are the little bumps on his claws. Draw small, draw big. Draw small monsters, turn the page, and draw the things. No.
What? No.
I do the girl, and that's all right. Oh, I need a second. This is where they go after the shoot.
Right now, no, you have all one page.
So, you want to concentrate on the female for a little while? You have to make it happen. You can see her apron. It's not a female, she likes to scurry, so you gotta get fast. Her heel pads are pretty much the same size. You look hard, sometimes you can see a little bit of red even on them. My slightly hinges sometimes, you can see a red dot. Crabs have a pretty complicated mouth. You got some outside parts, and then inside there there's another group of parts, and inside there there's another group of parts. So it's like there's different layers of mouth parts. When you get a crab right, she's got like a toe and a middle joint and another middle piece. Got hairs on their legs. It's getting a little complicated here. Get it right, you can probably see chin and legs. Sometimes it doesn't show all together. Another helper over there? Use that bigger cup if they keep falling out. You can just use that bigger cup to help us. Want to bring the big cup? And then just put them. Close this. Okay, let's go. Okay. Okay, we got we're gonna start this whole component using humans as our model. What I want us to do is measure heart rate. The way that we do that is measure what we call our pulse, but I don't want you to do it doing it on your wrist. This is one place where you can measure. There's something a little weird about measuring pulse. It makes you feel there's something right here that it's not really right here, right? The only thing you want to push your blood throughout all your organs. You're feeling this pressure wave of that blood passing through you guys. So the way you find your carotid pulse is not your thumb, but your two fingers sort of on your Adam's apple. This is the laryngeal prominent here. Find that, and then you push in a little bit deep. That's where you can use the palpate carotid pulse. I like to use beats per minute. How much per minute is your heart rate? Next, Mr. Allen. How do you feel about working the computer on this? People are gonna come up to you, and they're gonna whisper their pulse to you. Okay, everybody just get about their pulse. Can you come up and whisper your beats per minute?
Like 70.
Table two, if you haven't given your pulse to this energy yet, you can do that now. Somebody, you know, yours not working, you add it in. Yeah, no pedal system. This is useful. Males in the crab. This was our anatomical component. I am looking at these phenol numbers. Informative to look at raw data. My IDs look smaller than the male. But we usually do something in order to get a good feel for that. Take an average. So to take an average, pretty easy. You just go into the function. What to take an average of. Probably all y'all know how to do that. Go underneath it like this. Put in a function, average. So, okay. Yeah, it works. The average of that group or something. At 4.8. Take these, pick up the G arm. So this is in grams. Once you put in an average, you can just copy that. Boom. Okay, so let's compare that. 4.8 to 10.8. That's different. Okay, the average for the females, their mass is 4.8 grams. The average for the males is 10.8. When you're thinking about data, that is informative, even more informative than having your eyes have to go through all of this and kind of, what your eyes tend to do is kind of make an average, right? If you're a scientist, get an average like that, then that's a little easier. But there's another thing that's important when you're thinking about data, and that is the spread of the data, right? Each one of these numbers is not exactly the same as the average. Right? The average kind of represents them all, but not one of them fits right on the average. So the spread of the data in statistics is what we call the variability. In biology, we always expect variability. If there's no variability, you almost don't trust it. Like in physics, you measure a whole bunch of volts. Okay, you don't really expect a lot of variability. But in biology, you expect variability because we're all accustomed to how different biological creatures are. Like even if you're almost all the same age, we're all very different, right? So what we use as a measure of variability, one thing that we use is called standard deviation. So in Excel, it's easy. You can use find it like that. Standard deviation. mp4. As scientists, when you see an average, you always want to know the standard deviation. An average should never be quoted without a standard deviation. You don't talk about the average in one sentence, and in the next sentence, talk about the standard deviation. Talk about this as really more or less one number. So what's the average of the female mass of crab? So they have 4.8 plus or minus 1.6 grams. This always goes together. You don't want to put it in your notebook that average and standard deviation always stay together. Remind yourself that. About the average in one sense and the standard deviation in another one, it doesn't matter. These go together. Now we're comparing 4.8 plus or minus 1.6 to 10.9 plus or minus 1.5. All right, that lets you see what the spread was. Standard deviations of these is not too different. So the variability is kind of similar, even though the averages are quite different. And my gut tells me that the average male fiddler crab is significantly different. But scientists, I need to use the statistics to really put that down in the report. So next time we're together and working with numbers, we're going to use the student's t-test. So raise your hand if you use the student's t-test. Somebody, when we were doing that data a while ago, they said, is it okay if there's not the same amount of boys and girls? In the student's t-test, we measure N. It's part of the algorithm, right? In most experiments, there's never the exact same amount. Right now, there's a lot of press about testing the vaccine, right? They're not getting the exact same number of males and females. You just get what you can get. You get what phenotype you can get, right? We didn't have as many males and females. We don't say to ourselves, well, we can't do that in our past because we don't have the same number. No, you just do it and you mark down what is the N. The N is a sample number. This is the average. This is the standard deviation. And the N, grab 1, 2, 3. Here is 5. And over here, 10. N for females, 14. All right, I'm going to put this on Moodle, and we're going to work with this data together. We're going to use students t-test to test whether this is significantly different than this. We're going to use students t-test to test whether this is significantly different than this. So whereas males appear to be almost twice as large as females, when we look at heart rate, now look at that difference. Okay, 71.5 is a different numeral than 79.4. That's a different value. But is that significantly different? It's going to be close. In my experience, whenever the average is different, there's a chance it's going to be significantly different. But I can't exactly really make a prediction of whether these will be significantly different using the student's t-test because they're close enough to where it's not significantly different. When we use students t-test in biology, we set a p-value of 0.05. That's kind of like the significance value. You can do it at a different value. There's a way that we can make sure they were significantly different, but by convention, we almost always use p-equal 0.1. You said you used the student t-test before. Did y'all talk about the p-value? How do you explain what the p-value means? What is p represent? Yeah. It's a one term for p is probability. And one way to explain what does it mean to have a p equal to 0.5? It's so arbitrary that I made myself memorize it. This is a way to explain to someone else the next time you have it. What does it mean? It means that 95% of the data can be explained by non-random events. They fuse the point. 95% of the data can be explained by a non-random event. That's the power that statistics do. In this case, what's our non-random event? Our non-random event is that we're looking at one number that represents males and another number that represents females. Sometimes you do that. Sometimes you represent humans before exercise, humans after exercise. Sometimes it's humans that are less than 20, humans that are more than 20 years old. Something like that, right? That's a non-random difference. All right. The last thing that I wanted to talk about before we left was to go back to our superorganism. Using Mucomynax today. I wanted you to know about Mucomynax's role in the ecology of the estuary. You know how in Lord of the Rings, there's all these big dwarves and stalwart humans and sly elves, but they gave the ring to the smallest of the little hobbits. Sometimes the smallest gets ignored. Fiddler crabs are like some of the smallest organisms in the world. It's easy to think, what's talking about that? Kind of low. I think of them as the unsung hero of the marsh. Important as the littlest of the Fellowship of the Ring, the Hobbit. Hobbit had the least amount of ambition, so he was the one to carry the ring. Little crabs are more or less the farmers. Naturally occurring more. When you drive around the outskirts of Thibodeau, you go past a lot of sugarcane fields. What's it take to grow sugarcane? You need a farmer, a tractor, he plants it. Sometimes adds fertilizer, sometimes adds pesticide. The farmer's doing a lot of work to make that monoculture, make it grow every year. We make the grass grow in coconuts. There's no farmer out there, there's no tractor, there's no fertilizer, there's no pesticide. That's because like Mother Nature planted that grass. When you go and eat it to Grand Isle or the coconut, most of that grass is Spartina alternate. What was called that oyster grass? And who is cultivating that grass? It's a big monoculture. It's almost as dense as a sugarcane field. There's no farmer out there. No farmer. He's the Minax. And his little cousins, Ecologists. And their little cousin, Tennessee. These are the three fiddler crabs that you find on the Louisiana coast. They're all fiddler crabs. They all have a slightly different place that they live. You can kind of see like the beach. Ecologists like salt marsh. Like on the back of Grand Isle or back of Last Island, the back of Timberlake. Minax like more like the freshest salt marsh. So the closer you get to like a cypress swamp, there's still some salt marsh there. That's where you find Minax in the highest part of the marsh. What these guys do, they live in a symbiotic relationship with Spartina alternate. They are protected by living underneath the grass. And they pick little parasites off of the grass and they eat that, like kind of like a blue crab. Sometimes they can eat plants, but they can also eat little insects, right? So they pick off the insects. They are detrital feeders, and so they kind of filter stuff out of mud and make a burrow into where the Spartina grass lives, and so they bring oxygen down to the roots of the Spartina. And then they defecate, so they make feces that acts as fertilizer. So these guys oxygenate. They eat detritus. They fertilize. And they live symbiotically. Spartina alternate, it can grow without fiddler crabs. And fiddler crabs in our lab here, they can stay alive without Spartina alternate. But in the real ecosystem, there is the grass, plus the fiddler crab, and another fiber. Usually there's a muscle called the ribbed muscle. And you also find around the roots of Spartina. A lot of times you'll find a snail. It goes up and down the strands of Spartina. So there's grass, there's a crab, there's a mussel, there's a snail, and all of these work together. And nobody put them there. Like the wildlife of the marsh. And it's the marsh system, you find them all together. Now, the periwinkle is just this little snail. And at first, people thought that what he did on grass, as he climbed up the grass every day, I thought he was like grazing off of the grass. But now what they know that periwinkles do is they go up and down, and they leave slimes. Right? A snail leaves a little mucus trail. And really what they're doing is that mucus attracts a fungus. And now it's understood that what the periwinkle does is cultivates a little micro cultivar of fungus up and down spartina grass, and it goes back the next day and eats that. So what the snail is doing is not just as simple as eating grass, he's using the grass to grow a fungus that he comes back and harvests every day. That's pretty cool. Okay, we don't get out this early every day, but today we're going to get out early. So if you want to talk about honest fixing, you can stay. Or if you want to talk about the travel trip, you can stay. Or if you want to talk about the exam coming up, you can stay.